All right, so now we're going to talk a little bit about immunoglobulins. Specifically, what are the genes that code for immunoglobulins, code for antibodies? But then how do you get diversity of antibodies? How do you get different variable regions from one antibody on one B cell to another antibody on a different B cell to a third antibody on a third B cell, for instance? So we're going to focus in on that then. Okay, so antibodies and T cell receptors, those are the big antigen sensing things in our adaptive immune system. They can respond to a limitless array of foreign antigens. Virtually every antibody that's been found has a unique variable region. So no two snowflakes are alike type of situation. But only one of a limited number of constant regions, the whole IgM, D, G, E, A. Okay. So how do we get variation with a limited gene set? How do we get one part of the antibody that is varied, but a different part of the antibody is constant? That is what we're going to focus on. How do you get variation and how do you keep one part of the antibody varied, but one constant? That's our focus in this lecture set. So what we're really going to start also looking at is the life of a B cell. So this is going to be like the B cell's time to shine, its own little movie portion, okay? So we're going to look at this one at a complex series of events that takes place during the maturation of B cells, the activation of a B cell, and then the differentiation of a B cell from start to finish. And so I'm really going to start today and a little bit tomorrow going through a chronological look over the sequence of events in a B cell and their locations. Whether or not it's a B cell maturing in the bone marrow or the B cell then out in the environment when it encounters antigen or antigenic challenge out in some secondary lymphoid tissues and organs. So we're going to start off looking at B cell maturation, and that's going to be in the bone marrow. Then it was where this is going to take place. Again, you're going to have a hematopoietic stem cell that turns into a lymphoid progenitor cell that then turns into some B cell, pro-B cells, and then you're going to get those antibodies produced. What's going to happen here, though, is this process of maturation in the bone marrow is going to result in the generation of unique variable light domains and also variable heavy domains in those antibody structures. So you're going to get unique variable domains in both the light chain and the heavy chain. And all of this is going to be in the bone marrow before it sees antigen. So this is all independent of antigen before an antigen is ever seen. And again, remember, we always start off with generating either an IgM or IgD heavy chain first. That's what we're talking about here. You're going to end up at the end with a mature B cell that has membrane-bound versions, membrane IgM or membrane IgD, coming off of its surface. Okay, now that's what's going to, we're going to start focusing on a lot of this. Tomorrow, we're probably going to start focusing on some of these. Tomorrow, once you got a mature B cell is going to move into the lymphoid organs and tissues. It's going to move into things like lymph nodes or MALT, for instance, mucosa-associated lymphoid tissues. And then it just sits there. And if it never encountered its antigens, the B cell might die after about 2 weeks. But if it does, if it has antigenic challenge, if it sees its antigen, that's going to activate the B cell, and then it's going to cause it to differentiate, maybe into a memory B cell or then a plasma B cell, for instance. Typically, the first antibody it produces is IgM. Here's my pentameric IgM. But then it can further undergo activation into things like heavy chain class switching. So maybe it switches over to IgG or IgA or IgE production, or some other things called hypersomatic mutation and affinity maturation. We'll talk a little bit about this, but sometimes the B cell undergoes a mutational process that makes it better at binding to its antigen. But we'll talk about that here a little bit later. Okay. Now, setup for some of this though. Well, I'm going to first talk about this beginning things today. Okay, we're going to focus on the maturation in the bone marrow. So, central dogma of biology, flow of information to cell. You guys are probably all familiar with that. DNA is the blueprint for living organisms, for cells. DNA is information. It is then converted into RNA, which is then converted into a final functional product, usually protein. And the protein goes off and does a function. So that's just the flow of information inside of cells. The traditional view of this flow of information is that one gene in the DNA codes for one polypeptide, one protein. That was kind of the traditional view, is this segment of DNA codes for this protein. That's just the way it works. And it's also wrong. It's not wrong. It is fairly common for one gene to equal one protein. But the more modern view is that one gene does not always equal one protein. So for instance, some genes don't code for proteins at all. You have some genes in the DNA code for functional RNAs like a transfer RNAs, ribosomal RNAs, or microRNAs, which are involved in gene regulation a lot of the time. The other thing, though, is sometimes you have alternative splicing of exons and introns, which we'll look at a little bit here. But sometimes, do you guys remember how many genes we had that I said earlier in humans? Yeah, the current estimate is about 20,000 to 25,000. Depends on who you ask and what year you ask it. When I first started teaching, it was 25,000, and they've gone down to 20,000. They've actually reduced the number of what they think are genes. Do you guys know how many proteins we can make? They look at the human proteome, so all the different proteins in our cells. They estimate there's about 100,000 different proteins that can get made. And why is that? It's because of alternative RNA splicing. Sometimes you can have a gene. Do you guys remember what the difference between exons and introns are? Yeah. What are the exons? The expressed or coding regions. And the introns are the non-coding regions that are cut out during RNA processing. But if you look at gene, for instance, if this is my RNA molecule, maybe this portion is exon 1, and then you have intron 1. But maybe then this is exon 2, intron 2, exon 3. And again, maybe intron 3, and then maybe you have an exon 4. Okay. Now, what happens during RNA processing is typically the introns are cut out, cut, removed, and then exons are stitched together. So at the end of RNA processing, you might have a shorter RNA molecule that is exon 1, 2, 3, 4, all joined together, and the introns being removed. And that's what happens sometimes, but sometimes you get alternative splicing. So instead, you get exon just 1, 2, 3, or you can get exon 2, 4, for instance. Sometimes you actually cut out some of the exons as well and stitch them together differently. That means that one gene could code for protein 1, 2, and protein 3. So alternative splicing can sometimes result in one gene equaling different protein. And maybe all those proteins are functional and do different jobs in the cell as well. Okay, so alternative splicing can actually increase the diversity of proteins we can make as well. And then there's antibodies, which is another version of where one gene does not always equal one polypeptide. In this case, it's where many genes can sometimes equal one polypeptide. So it's almost the opposite of alternative splicing. Alternative splicing is where one gene can equal many proteins. One gene equals many proteins. Antibodies are going to involve many genes equaling one protein. Okay, so the enigma has basically like, how can you get all these variable regions? And then how can you have a variable region versus a constant region of these heavy and light chains? So there was two competing theories early on during immunological research, what is called either the germline theory or the somatic variation theory. And the idea behind this is the germline theory is that how do you get different variable regions? Well, you just have a huge number of immunoglobulin genes in the genome, and then they're mixed and matched to make one variable region. So what I mean by that is you might have gene 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. But then sometimes you use 2 and 3 to make a variable domain, variable portion of light chain. Maybe sometimes you use 5 and 8 or 7 and 10. You mix and match genes to make a variable domain, and that could get you increased variation. Does that kind of make sense? And we're going to go through this. The other theory was what was called somatic variation theory, that there isn't a huge number of genes, that there's a small number of genes from which specificity and variation is generated by directed mutation. Just take the genes that code for variable heavy domain or variable light domain and just randomly mutate them, is this idea. So these were the two competing theories for how you got antibody diversity early on in research. Any of you guys happen to know which one ended up being the answer? It's a trick question, both of them. Turns out both are right and are part of the process, which we'll talk about. Okay, so what was some of the early research? 1965, researchers, last name of Dreyer and Bennett, they proposed that there must be hundreds or even thousands of different genes for the variable regions, the variable domains of both light chain and heavy chains, but only one constant region for the light chain and only one constant region for each of the isotypes of immunoglobulin. What are the isotypes again? The classes, the IgM, D, G, A, E, the different classes. So you have 5 different constant regions for each one of the 5 isotypes of heavy chain. And so what they were doing was, this was basically proposing germline theory, where they could mix and match genes to create unique versions of variable domain portions. And then in 76, a group of Japanese researchers, Tanagawa and Hazumi, provided actual experimental proof of that then. And they showed that there was a large number of genes that could create the variation, but also that there was DNA recombination. The DNA was being cut and restitched together at these antibody genes of the variable domain of the light and the heavy chain. What they did is they did a southern blotting technique, and they compared light chain genes from mouse myeloma cells. And myeloma is actually cancerous B cells, is what a myeloma is. But these are ones where the B cells have already gone through antibody maturation, for instance. So they were already producing antibodies. So antibodies are fully developed versus mouse embryonic lines, which B cells haven't even been made yet, but it still has the genes for the B cells. They just aren't B cells. Does that make sense? So prior to B cell maturation. So the idea is after B cell maturation, before B cell maturation. And they use a southern blotting technique. Now, southern blotting is an old school technique that not a lot of researchers use anymore because there's more modern ways to do it. But this was back in the 70s. I have done southern blotting before because apparently I'm that old. Anyways, the idea with southern blotting is you isolate DNA from cells. So you isolate all the genomic DNA from cells. Then you cut it with restriction enzymes. Restriction enzymes are just enzymes that cut DNA. So you take that DNA and you cut it up into fragments. Now you have these smaller fragments, and you can use gel electrophoresis to separate out the DNA fragments by size. You guys have, I think, all done gel electrophoresis before, correct? And the idea is it separates, you load the DNA onto the gel. DNA is negatively charged. So you put a negative charge at the at the top, positive charge at the bottom, and that forces the DNA by the electrical charges through the gel. But long fragments don't move as far because they have friction with the gel. Shorter fragments will move further because they have less friction with the gel. So you get long DNA, shorter DNA, separates out those fragments by size. They denature the DNA and they transfer it from the gel to a blot. And the way you do this is literally you take the gel, you set it on top of a piece of blotting paper, and then you apply electrical charge down underneath it as well. And by the force of gravity and that slight electrical charge, you bring the DNA down from the gel onto the paper. The blotting paper is also positively charged, which interacts with the negatively charged DNA and holds it in place. But you keep the same banding pattern. If the DNA fragment was here in the gel, it's there on the paper then as well, the blotting paper. Then you prepare a radiolabeled mRNA probe. So we make messenger RNA that's complementary to your DNA of interest. So if you have a particular gene and it has a sequence of ATCG, we make a then RNA probe that is complementary, I can't remember what I said. Anyways, it's a complementary sequence. But we've used, the RNA is actually assembled and made by using radioactive phosphates, P32, which has got alpha radiation. So not particularly strong, a couple pieces of paper can block it, so it's still fairly safe to work with. Until somebody in your research lab spills P32 into your microcentrifuge, and you have to quarantine the microcentrifuge for 6 months because you have to let the radiation decay. Has a half-life of about a couple weeks, but it takes, that actually happened. It wasn't me, it was somebody else in my research lab. But anyways, they were getting the tube out and boom, just dumped it into there. We couldn't. All right, anyways. But what you do is if your RNA probe is complementary to the DNA, it binds it. So wherever your gene of interest is, your RNA probe binds it, you wash off any excess probe, but if your DNA fragment that was complementary to is here on the gel, it binds to it and you get a radioactive signal. You can actually just put a piece of X-ray film over the top of it and see where your banding patterns are. Questions about that? It's pretty neat, it's fun. Okay. Now, what did they find? In differentiated cells, the myeloma cells, the ones that are mature B cells, DNA rearrangement had occurred. So what they found, for instance, is that if they did the germline DNA, they did a probe. They did one probe against constant region. They did one probe against light region, for instance. The light chain, heavy chain, light chain is what I should say. In the germline DNA or embryonic DNA, the probes were very big. They were very long, is what they had found. But then when they did the DNA from the myeloma cells where they had undergone, they were making antibodies, they were much shorter. What they found is a large chunk of DNA had gotten deleted out of the genome of these cells. And really what had been happening is that the DNA was getting cut, stitched back together to create unique light chain regions or unique heavy chain regions. So the DNA was changing in size. Questions about that? Okay. So they were undergoing rearrangement during B cell development. Think of this as a cut and paste. It's actually mostly just a cut mechanism. Actually, yeah, you know what? That's wrong. I said cut and paste. It's really, honestly, they've got a couple portions of DNA and they cut out everything in between it and then stitch the two together. So it's actually, yeah, cut and paste is wrong. I don't know why I got that up there, but it's cut and just cut. And I'll show you that here in a second. But this is the primary mechanism that drives diversity of antibodies or immunoglobulins. And also, this is the only known site of DNA-specific recombination and rearrangement in vertebrates. Only antibody genes and T cell receptor genes do this. So it's only the immune system and adaptive immune system where this takes place, where you have recombination events DNA gets cut and put back together in a site-specific or DNA sequence-specific manner. It's only in vertebrates that this happens with an adaptive immune system. Okay, so let's go through a little bit of what happens. Okay, again, real quick, I know we just went through it, but again, with antibody structure, you have the constant domains versus the variable domains. You have, again, 2 possible light chains, kappa or lambda. You have 5 heavy chain isotypes, M, D, G, E, and A. Okay. But the VEG chains have a variable light chain domain and a constant light chain domain. Most of the heavy chains have a variable heavy domain, 1, and then a certain number of constant domains. For instance, IgG has usually constant 1, 2, and 3. Okay. So you have these constant domains versus variable domains. Now, if we look at then the genes that code for either light chain, lambda light chain, kappa light chain, or the heavy chains. Light chain genes are on human chromosome number 22. Kappa light chain, lambda light square light, kappa light chains on human chromosome number 2, heavy chains on human chromosome number 14. So they are on different chromosomal numbers. They are separate from one another. Okay. And they're encoded by these big, what are called multi-gene families. In a multi-gene family, this is where you just have several genes or gene segments of related function clustered together. So they're all in the same portion of, for instance, lambda light chain is all in this one big area of chromosome number 22. So multiple gene families where these genes are just clustered together next to one another on a chromosome. During B cell maturation, gene segments are going to get rearranged and then brought together to form a then functional immunoglobulin gene. And we're going to take a look at how this kind of works and what is the multi-gene family involved here. Okay. In a light chain, light chains are going to contain multiple what are called V, J, and C regions that are then brought together, and collectively, all 3 of those regions make up the variable domain of a light chain. So you put V plus J plus C equal, for instance, the variable light domain. And just that portion, just the variable light domain, the constant is not involved with the gene rearrangements. I'm sorry, I got it wrong. Guys, sorry. The V and J regions make up the variable domain. The constant portion just makes up the constant light domain. I got ahead of myself there. Sorry about that. So V and J segments are brought together. These make up the variable domain. This is just makes up the C region, makes up the constant domain. Sorry about that. But you have multiple V segments, multiple J segments, and then usually only one constant segment. So with the V and J, again, there's going to be multiple V's, multiple J's, but only one constant for light chain. Heavy chains are going to contain multiple what are called V segments, multiple G gene segments, multiple J gene segments. The V, D, and J segments, those are going to make up the variable constant domain. Whoops. So those are going to make up the variable domain, whereas the constant region is the constant domains. Okay, I got these diagrams down at the bottom. There's a lot going on here. But remember, with constant, with heavy chains, you can have different isotypes. And by the way, this is the constant region for the mu or IgM isotype. Delta for IgD, IgG3, IgG1, IgG2, IgG2, IgGE, and IgGA. That's their arrangement. So these are the constant domains. Okay. But then you have multiple V segments, multiple D segments, and multiple J segments. that are used and mixed and matched to make a variable domain. All right, so what's this look like in a bit more detail? What I'm showing you is kind of the germline gene segment organization of kappa light chain in mice. Why am I switching from mice to from humans to mice now? Just mice are more studied and we know how this works a little bit better in mice. So we're going to use that as an example. In the germline DNA, this is the embryonic DNA for B cell maturation. What you find is there is V segment 1, V segment 2, V segment blah, 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 all the way up to V segment number 91. There are 91 V gene segments in a row. So it just goes V1 through V91. So this is supposed to be this dot, dot, dot is just a lot of other ones here all the way up into V91. Does that make sense, everybody? Okay, I just can't fit all of those onto the slide. Remember, light chains have V and J segments. And so the other thing it has is J segments over here, and there's 5 J segments, but this one doesn't count. This one doesn't count because that psi is a symbol for a pseudogene. It's mutated in some point in its evolutionary history and it's no longer functional. A pseudogene is a false gene or a mutated non-functional gene segment in essence. So you just have 4 J segments that count 1, 2, 3, and 4. And then again, in kappa light chain, you got one constant segment, kappa, constant kappa domain. This is big. It spans about 3.5 million base pairs of chromosome number 6 in mice. That's a big chunk of chromosome number 6 is just antibody genes. Lambda light chain in mice. It only has 2 V segments. And then it has 3 functional J segments, so it's got fewer. It actually has a couple different C segments, but don't worry about that too much, and some pseudogenes mixed into there. So lambda light chain is not quite as diverse as kappa light chain. And you also see some of the V and J segments are interspersed a little more funky. Okay, so lambda light chain is a little bit odd. Okay. Again, in both the light chains though, kappa or lambda, the V and J segments are going to come together to form the variable domain, the variable region, whereas the C segments are going to form the constant region of the light chains. And then heavy chain, it has V, D, and J segments. Again, all of those are coding for the variable region, the variable domain of the heavy chains, whereas the C segments code for isotype, IgM, IgD, here's the IgGs, IgA, and IgA. In mice, heavy chains has 101 V segments, 14 D segments, and then 4 J segments that can rearrange. But at some point, a V has to come together with a D with a J. You have to have what is called VDJ joining, and all 3 segments contribute to the variable domain of heavy chain. Questions about that so far? Okay. Let's get to the point. And so let's get to the point. Now that I've kind of gone through the gene reorganization, let's now actually hit the rearrangement stuff. So how do all of these gene segments add up to a antibody? And how does then all these gene segments equal a B cell getting a unique version of an antibody and a different than other B cells? So let's go with kappa. Let's go with kappa light chain. Again, it has many variable segments, 5 many V segments, many joining segments, and then 1 constant segment. So this one's a little easier to follow along with. And I showed you that germline DNA kind of arrangement before. But during B cell maturation and development in the bone marrow, a random, and it is random, a completely random V segment will be brought together with a random J segment. And this is done by DNA recombination. So what's going to end up happening is the first step in making a light chain is what is called VJ DNA rearrangement or VJ joining is the other term for that. V segments and J segments are joined together to form a functional variable domain of the light chain. So in this case, I'm theoretically using V segment number 23. Remember, there's 91 of these guys. I could have used V segment 1. I could have used V segment number 91. I just randomly, 23 was chosen. So V segment number 23 comes together with J segment number 4 over here. Okay, and then you have VJ joining. So that V segment gets directly joined to the J segment. Remember, and this is going to make then the V chain codes for the first 97 amino acids of the variable domain, whereas the J chain codes for the last 13 amino acids of the variable domain. Remember, I said there are about 110 amino acid domains. Now, remember, this is taking place in the DNA. So all of this is germline DNA in my diagrams here. What is happening is literally you're taking the V segment DNA and joining it together with the J segment DNA. But when that happens, you have to cut the DNA here. You have to cut the DNA here. And when you cut that DNA, intervening sequences are permanently deleted out of that B cell lineage. So if you use V segment 23, you permanently delete out V segment 24 through 91. When I use J segment 4, I deleted this one, and this one permanently out of the DNA. The DNA gets cut. That DNA fragment is lost and degraded. And the two other DNA fragments are stitched together. So I cut here, and I stitched them together. And this is all DNA recombination. Everybody okay with that? But now, so all other cells in your body, skin cells, embryonic cells, muscle cells, they've got the full complement of all of these antibody genes and they're carrying them in their genome, but they never went through the maturation process and the B cell maturation. They didn't get the signals to do this. It's only in B cells that this DNA rearrangement step happens, okay, for antibody genes. For kappa light chain, that's it. You're done with the DNA rearrangement. All you got to do is do it together, V and J, done. VJ joining, DNA rearrangement, you finished it. So you now have a recombinant VJ region. And what's it do? It undergoes transcription. So now we go from our rearranged DNA, it goes through transcription. So now the recombinant VJ region and the C region are then transcribed into a messenger RNA molecule. So this is my primary RNA transcript before RNA processing. So it's a pre-RNA is the other way to say this. So this step right here is a pre-mRNA. Okay. But then it goes through processing into a final messenger RNA transcript. And the RNA splicing will remove introns, but it also will cut here and here in the RNA and bring together by RNA processing VJ and the C segment. So now I have an uninterrupted final messenger RNA coding sequences. And if there were any other J segments left in there, they get cut out during the processing. So this was still in there, it didn't get deleted because we used the fourth one. So this guy was still there, but it gets cut out by the splicing event. Okay, and you guys comfortable with introns versus exons? I know we kind of already talked about that, but I got it on here if you need it. Again, introns are the non-coding regions, exons are the coding regions. Okay, all right. But then what ends up happening is you got your final messenger RNA, That is then turned in by translation into an antibody protein, the light chain. Questions about that?
My question is, what time in the population happen is when the kid is in the.
Oh, no, all the time, all the time. Yeah, you're constantly producing new B cells in your bone marrow. This process is always happening in your bone marrow throughout all of childhood, adulthood, every time. So you're constantly making B cells by this process in your bone marrow. Okay. So, questions about that? We should have one question. I've had all those little black boxes labeled L in every single one of these diagrams so far. What the heck are those guys? You might have, so whenever you're seeing diagrams, stuff aren't on there by accident. These diagrams are something you want to study and use while you're studying. You guys probably heard pictures worth 1,000 words. So the diagrams are ways to go through this and understand it a little bit better than all of my text in many cases. So some of you might have wondered what those little black boxes with the L's are, and what they do is it a gene segment that codes for something called leader peptide. And you might see here during the RNA splicing, you cut out the intron that was right here. This is supposed to be an intron, and you join the leader peptide right onto the very beginning. The spliced processed RNA moves from the nucleus to the cytoplasm and the ribosome by translate, and then it gets translated. But that N-terminal leader sequence is called a leader peptide, because what it does is it directs, it gets made first because it's at the beginning of the protein. But this will actually, leader peptide sequence directs that new protein being made to get made as a, it directs the ribosome to go to the rough endoplasmic reticulum. The ribosome binds with the rough endoplasmic reticulum and then makes the new antibody protein into the rough ER. The whole thing here is bound versus free ribosomes. This is probably the last thing I'll talk about for today, and then we'll pick back up with this tomorrow. Polypeptide synthesis. Remember, you have a messenger RNA binds to a ribosome in eukaryotes in the cytoplasm, and it starts getting made. Polypeptide synthesis translation always starts in the cytosol, the cytoplasm, and by default will finish there. So typically the default state is a free ribosome making proteins in the cytoplasm. Everybody comfortable with that so far? Unless there's a leader peptide. So synthesis will finish in the cytosol unless the polypeptide, the protein being made, has something called a signal peptide or a leader peptide at the N terminus of the protein, the beginning of the protein. The signal peptide is a very particular sequence of amino acids at the beginning of the protein. So for instance, here's my ribosome bound to a messenger RNA in red. It's making a new protein. It just started. And these little spheres are supposed to be my amino acids. But the very beginning of this particular protein being made, it's got a specific sequence of amino acids. That's my signal peptide or a leader peptide is what it's sometimes referred to as. And if this is present, it is bound by a protein called signal recognition particle or SRP. And what this job does, if it's activated, it causes my ribosome to become, instead of a free ribosome, it causes it to go to the rough endoplasmic reticulum and become a bound ribosome. It binds onto the surface of the rough ER. And if it binds onto the surface of the rough ER, it makes the protein through a pore in the rough ER, and it actually makes the new protein into the interior of the rough ER. So it makes it inside of that particular organelle. So the SRP causes the ribosome to dock with the rough ER, becomes a bound ribosome, and then the protein is synthesized through a pore in the rough ER, and it ends up inside the rough ER. Now, why is that important? What kind of proteins are made by bound ribosomes and are in the rough ER?
to make this exocytosis?
Yes, so they're gonna get, usually proteins in the rough are either gonna get secreted, often by exocytosis, or the second fate is also sometimes membrane bound. So typically proteins made in the rough ER, if they're made in the rough ER, they're either proteins that are secreted or membrane bound. And what are antibodies? Sometimes membrane bound and sometimes secreted. All right, questions about that light chain rearrangement? Okay, by the way, heavy chain rearrangement, just to go through this real quick. Now I'm gonna go back, I'm gonna pick back up with this tomorrow. But it's the exact same thing, except for you have now, instead of just V and J segments, you got V, D, and J gene segments. So this is a good spot to take a break, actually. But we'll go through some of this, pick back up with this tomorrow, because it's going to be the same thing. So it's a nice review. All right. For tomorrow, content worksheet. I think I need to edit one question. So even while it's up, don't start it right away. And I'll get that one question that's up there changed. Okay. I think I missed by about 10 slides.
Just wait to do it, so you email us that.
Yeah, it shouldn't take long.